Okay, I'm Gemma Arnau from CIRAD, Yam Geneticist, and working on work package 4. And why accelerating breeding of Yam Dioscorea alata through genotyping by sequencing? Breeder yam is a food crop of great potential. It's second to Dioscorea rotundata in the volume of production, has an ideal potential, a long storability of tubers, it's easy of propagation, and has an early growth vigor for weed suppression. The most important criteria of breeding are ideal potential, tolerance to anthracnose, a fungal disease caused by Coledotrichum, and tuber quality, which depend both morpho-agronomic traits and physicochemical characteristics. Breeding for tuber quality, it's a long process because there are many traits. Several are strongly influenced by environmental factors. That's why our objective is the development of molecular markers, SNPs, associated to genomic regions that determine tuber quality, useful for marker-assisted selection. Genotyping by sequencing, with the development of sequencing genotyping technologies, has facilitated the development of high-density genetic maps with codominant markers, facilitating fine genetic studies as the detection of genomic regions associated to agronomic interest traits. We construct the first high-density map of the alata as a part of Africa Yam and Cabalbio projects. To identify QTLs for tuber quality, morpho-agronomic tuber quality traits, some external has tuber size, regularity of shape, skin and hairiness, and some internal has the flesh color and the growing of the flesh. As a part of Africa Yam project, we developed two biparental populations that we phenotype for all these traits. About 300 hybrids, 9 replicates, 2 blocks, and 2 years. This trait, it's difficult to select the regularity of tuber shape because it's strongly influenced by soil conditions. You can see here the distribution of two progenies. The population A in the left, and the population B in the right. And this trait is a polygenic trait. And we observe some transgressive hybrids with values either than both genitors. You can see the first pictures, hybrids with very malformed tubers. And we detect 5 QTLs explaining 90% of the total phenotyping variance. 3 of these QTLs were validated on a diversity panel of 28 varieties. You can see one of these loci, and for this loci, 3 different genotype classes were observed in the diversity panel. And all the varieties having a nice and regular shape are homozygote for the allele A. And the other varieties are heterozygote or homozygote for an alternative allele, which is the unfavorable allele. In conclusion, the locus identified are not specific of genitors, and the marker level will be useful to select the best parental combination and also promising hybrids in segregating populations. We are working also on the identification of QTLs for physicochemical and textural traits, chemical properties, dry matter, starch content, proteins and sugars, and textural properties, hardness, moldability, cohesiveness, and springiness, as a part of RTV Food project, in collaboration with INRAe from Guadeloupe and CNRA from Côte d'Ivoire. To be in measure to phenotype the biparental population, we developed first on a diversity panel on 27 varieties, about 174 samples for chemical analysis and 81 for textural analysis. And for these 27 varieties, we analyze chemical properties at Laboratoire Tessier, textural analysis using a texturomet TPA in Guadeloupe, and the spectra were generated with the NIRS NRA system from INRAe. And with the NIRS prediction models developed, it was possible to phenotype both populations about more than 1,000 samples. And preliminary results for starch content show the distribution of this trait is normal, that it's a polygenic trait, and 2 QTLs were detected, explaining 34% of the total phenotypic variance. We validate 1 QTL. You can see here in this picture that all the varieties with either starch content are homozygote, that is lossy, and the varieties with lower starch content are heterozygote. With thanks to different contributors from CIRAD Denis Cornet, Fabien Cormier, Eric Maledon, and Eline Oudol, from CNRA of Côte d'Ivoire, PhD Emmanuel Eonou, who came 2 times 6 months in Guadeloupe to work in this research, and Dr. Amani Kwaku, and from INRAe Guadeloupe, Lucienne de Fontaine and Karine Marie Magdalene, thanks also to following projects that made this research possible: Cabal Bio, Africa Yam, and RTV Food. Thank you.
Thank you, Gemma. And thanks for the effort of speaking English. I know it's not an easy exercise. Thank you. So is there any question? I don't see any in the chat. Is there anybody who has a question for Gemma? It was a really a synthesis of the work done in Guadeloupe together with CNRA.
I have a question.
Yes, thank you, Didier. Yes, please.
Good afternoon, everybody. Thank you very much, Gemma, for your nice presentation. I observed that you only presented result of star content.
Yes.
And, I want to know if you plan to analyze other biochemical traits. I think about polyphenol, for example, known to be involved in bitterness or in texture properties of RTB.
Okay, we intend to analyze the 4 traits I explained before. There are, of course, many other traits that will be analyzed later, but for the moment, we select these traits because there were techniques to analyze, easy to analyze, and other traits has polyphenols are difficult and when you focalize on these traits.
Okay, thank you.
I see two questions from Dominique, one from Michael and one from Astrid. So I will start with Dominique's questions. Could you specify the strategy that could be put in place within RTB Foods to test these varieties in African conditions with local consumers in Africa? Do you have any perspectives for this?
Okay, on the 27 varieties, the objective was to develop the nearest. Okay, and we include textural parameters to be measured to analyze quality of pounded yam. I think it's the more appreciated form of the consumption. And we have moldability. There are traits that it will be possible to analyze in the progenies. These traits, I think, are the important traits for Africa.
And so there is no plan in the near future to have tests or assessments with consumers in Africa?
Sorry, the objective of this work is to develop, to measure some traits, important traits. Mm-hmm. For that we develop it. That doesn't mean that, of course, the new hybrids selected from must be tested, but it's just one test to select some important traits.
Okay.
From founded and boiled yams for both.
Okay. Second question, what would be your needs in terms of high throughput screening of the populations? What are the most urgent needs that you would like to address with high throughput?
Yes, as I say, there are some, we are selecting for some quality traits, morpho-agronomic traits, as the tuber shapes, regular shapes. Some are easy to select for inbreeding, but others are difficult. Sometimes we can spend plenty of years and has the regularity of tuber shapes, and it's for that for this sort of traits, I think it's very interesting to have methods to easier select the hybrids. Others are not necessary. It's just the observation, the classical breeding. It's enough, but for some it's particularly difficult.
There are two questions from Michael and Emmanuel who are quite similar. Michael, do you want to ask your question to Gemma directly regarding the correlations, the good predictions?
Yeah, thank you. Thank you, Glantine. Gemma, very nice. Thank you for your presentation. So I understand the NIRS was used to, when you say you phenotyped the 1,300 individuals, I assume that was with NIRS. And I'm particularly curious about how good was the correlation with the texture measurements.
Sorry, I didn't understand. What is the correlation with what? I didn't understand.
Especially, well, the measurements that you did, you phenotyped after you got the NIRS equations trained, you then phenotype around 1,300 individuals, right? So I'm imagining you phenotype them with NIRS, but also you also phenotype for their traits to see correlations, or you've only done the NIRS for now?
Okay, we analyze to develop NIRS. The number of samples it was 7 varieties, and it's not too much sampled because a total of 1,074 samples for chemical and 81 for textural, but it was possible to obtain good equations and sufficient to use for phenotype the progenies. Better equations, I'm not a specialist. This work was realized by Denny and colleagues from INRAe, but the equations were good for all the chemical traits, excellent, and less for textural. But Denny Cornet is there. Maybe can speak with for textural. He developed other protocols of the prediction, and that's okay. It's possible to. It will be useful for the moldability, for the bonded threads. Interesting also. Sorry.
Denis?
Maybe Manuel can.
Yes, you want me to add something? Sorry, Denis Cornet. So I made the analysis on near spectra. So to answer one of the first question I heard, there is no clear correlation between chemical compounds we measure and texture attributes. There is some link, some correlation. So it's always linked to, but not sufficient to explain texture. In my comprehension, texture is a multi-trait attribute, and there is some compensatory effect and some threshold effect, and it's very difficult to link few compounds to global texture or quality attributes. On the question, can we use this calibration to predict texture attributes on biparental population? I will be a bit more cautious than Gemma. We made some calibration on 81 samples only, and calibration are very good. We made some validation on the same. Sorry? Okay, but I think the model are not very robust because of the small number of samples. And if we want really to apply it on a broad scale, we will need to make some external validation with maybe another year of observation. So for texture, it's still quite difficult to predict with a good insurance. So yes, let's say that we need one more year of external validation to be sure.
So can you, Denise, can you answer the question from Emmanuel directly? Can you confirm the parameters that worked well with NIRS prediction?
Oh, during the calibration and the internal validation, we had very good result for all parameters cited by Gemma, except hardness. But for cohesiveness, springiness, moldability, global quality, we always had very good performances. But most of these models are classification models, not regression ones. So we are not quantifying the traits, we are making some class. For instance, hard and soft, moldable, not moldable, good quality, bad quality. And for this classification, the accuracy was always higher than 80%, reaching sometimes 100%. So we had very good results, but on a very small number of samples.
Thank you, Denis.
Thank you. So the question from Ashrat is regarding QTL validation. Can you explain the procedure followed for QTL validation?
Okay. Okay. The QTL detected in population must be validated, and to validate, you use an external sample. In this case, you use a panel of diversity that was previously analyzed for all these chemical threats, starch content, It's the panel that you use for the developed NIRS. You had the data, phenotypic data. Donc, to validate, it needs to have the phenotypic data from a panel and also genotyping data. And you have both. And the validation consists to see if the marker identified during the QTL process, it's alleles, there is a diversity also in the panel for this allele, and to compare the genotypes observed in the panel with the phenotypic data of the panel. It's a correlation analysis. It's just to see if genotypic data is correlate to phenotypic data. It's clear?
Asrat, is it clear? Does it answer your question?
Yes, yeah. Thank you, Gemma. I think it is good explanation. I thought you have already converted those QTLs to some assay markers, and then you did the on the other. I think it's okay. I'm fine. Thank you.
There's a question from Jim. I am not sure whether it has been answered or not yet. Did you also conduct direct phenotyping for textural traits?
Sorry, can you repeat?
Did you also conduct direct phenotyping for textural traits?
Directly phenotyping of the panel, of course, for all these traits I was speaking about, but just on the panel diversity, on the 27 varieties, not in progenies. That's what's the idea of developed NIRS, and to be able to use sample method to analyze progenies, 1,000 samples, because textural analysis is it's a very long process. To do that on 27, we spent 1 month with PhD Emmanuel Leono, who came to Guadeloupe. It's a long work. And the interest is to have a more sample technique to analyze big numbers of hybrids.
There's a general remark from Michael, a question, let's say, which I think applies to the protocol developed by Denis on the good and bad genotypes. So the question is would a qualitative score, like was mentioned, good, bad, hard, soft, be enough to select material? Or do we need more quantitative data? So I'm not sure whether Denis wants to answer this question.
Okay, I think it's kind of a two-speed answer, because identifying the key traits driving texture and quality is very difficult, and it's a very long way. This is the heart of RTB food project, of course. We looked to kind of shortcut that could be useful for breeders. Of course, it is not a gold rule, but I'm sure it will be complementary to the classic approach of RTB food, in the sense that breeders don't really always need to understand what is driving quality. If they have tools that can discriminate good and bad, it's a first step, and it's maybe just sufficient to make the screening. So this was the idea behind the good-bad protocols and study. And I'm sure it is not sufficient. For instance, if we want to go to marker-assisted selection, or if we really want to understand how environment or practices play a role on quality, we need to go further. We need to go through each step of the RTB food project. But in the first attempt, we can already screen our genotypes only with this knowledge, good and bad. And this knowledge is for sure a good evaluation because it's coming straight from the stakeholders. With the database that people are giving me, we only sort genotypes based on what they know, good and bad genotypes. And we look if we are able to discriminate them with NIRS. And if we are, it means we have a first tool to start. It's not sufficient, but it is a good start in my point of view.
Thank you, Denis.
Hello.
Yes, Emmanuel, please.
Yeah, I just want to add to what Denis just mentioned. Actually, in the work package WP3, what we are trying to do, there are some difficult traits like that textural properties to predict. So we are looking for a way out to be able to help the breeder to quickly screen. And that is when that method of selection of classification to good and bad just coming. So it's a very good step. And then we are sure from there we can be able to move ahead from the classification. Thank you.
Yes.
Fabrice, yes, please.
Yes, just to complete, thank you, Emmanuel. Just to complete Denis and Emmanuel, this work is a first step for classification, and it will be complete by the work being done by Christian on cell walls investigation. So we try after to have something more robust between classification and more understanding of what is really involved in the hardness or other texture property. But the first step, we choose to do a classification because we did not have any other way to do it, because as Denis says, it's very hard to do a correlation between texture parameters and chemical parameters, and it's very hard to do directly near infrared linked to texture in a very, very efficient way. So for screening, we found, and thanks to Denis, with a learning approach, that it was more efficient to do classification, as long we know bad and good varieties. So this is based on knowledge before to build the model. But once the model is built, we can use it.
Thank you. Gemma, do you want to say something? There is another comment in the chat box.
No, thank you.
Okay, good. So there's another question or remark from Michael. I will try to translate if I understand well. Considering that shape is critical for acceptance and that soil conditions is a strong covariate, could we test the SNP alleles under different soil conditions, maybe to come up with recommendations for growing a variety in a specific soil environment and so on? So the question is,
yes. Yes, the interest is the shape is changing with the soil conditions, and it will be possible to use the marker to select for the shape in all the soil conditions. It will be not necessary to be very the work of the soil. It will be easier to select. That it's the objective of to have molecular marker for this difficult trait.
Michael, does it answer your question?
Maybe. I mean, that's the ideal situation, that the SNP is strong enough that it's independent. It overcomes the the growth conditions. But my understanding would be that that in spite of the selecting with the SNP in the particular soil conditions, it will still give the irregular shape. But maybe not. That's what I was asking. If kind of that's the that's the situation.
I can add something. This is Hannah. Hi everyone. This work was done on the biparental populations in one environment, but we have also the same work which is ongoing on a panel of diversity grown in 3 different environments, and which means also different conditions of soils. And we are also looking at the same traits in this panel. So we will, the idea is to see if we can find the same markers on this panel.
And just what is interesting is that the SMP were validated on the diversity panel. That means that are not specific of the genetic only the genetic fund of the genitors of the there are genes that are also in other varieties. It's for that I think that of course for quantitative traits has to change. The trait depends on not only one loci, but in several. But we need to select for not only one marker, but several QTLs, of course. But it's good because it was validated, and we observed that all the varieties with nice shape were homozygote at this loci. That means that it's a nice, interesting loci. I don't say that it's with just one locus, it's possible to select on it select for tuber traits, but it's encouraging. It's, I think, interesting.
Dennis, you have raised your hand. Do you want to say something?
Oh, sorry, it was long ago. It was just to say, as Anna said, that within Africa YAM2 project, we are testing on different environments with very different type of soil, vertisoil and feral soil, with different level of clay and so on. And we will have a good idea, I think, in 1 or 2 year maximum of the stability of this SNP on tuber shape.
And there's a directly aligned question from Elizabeth. At this stage, can the markers be used for screening yam in Africa? It's a broad question.
Okay, for textural and chemical traits, we are not finished. We are just beginning to QTL analysis. We have one marker for starch content, which was validated this year, but we are working on. But for morphoagronomic traits, the work that was conducted on Africa YAM Award, do we have some QTLs for each trait? I think they can be used for Africa, of course, because the traits are the same, and regularity of shapes, it's an important trait. Hairiness, the presence of rods in the tuber, it's a bad thing. We look for tubers with not hairiness. I think all these traits are common on Africa and everywhere.
Thank you. If there is no more question, I maybe have just want to conclude. What are the next steps in the collaboration with CNRA? Do you still plan to work together? How do you collaborate, or is it now finished?
For the moment, we must finish all this work of QTL analysis. I think the collaboration was very nice. it's important to be in measure to find cocktails for textural on ponded yams, but for the moment, I think concerning me, I'm working on QTL analysis with data obtained for the moment.
Okay. Is there any last question for Gemma? Yes, Denis.
No, we still contribute. We will still collaborate with CNRA in near future. We just submitted CIRAD grants to be able to host a CNRA scientist at CIRAD during a few months to try to train them on different SOP and different phenotyping tool in order to be able to implement that at CNRA in Côte d'Ivoire on their collection. So the collaboration will continue also on other traits, like color oxidation and so on.
Okay. Well, thank you. Thanks, Denis. If there is no more question or comments, I would like to thank Gemma for this impressive and very promising work. And as we can see, it's a teamwork. Lots of different speakers participating in the discussion, which is really, really nice. And it's also a good example of project complementarities, because as you can see on the slide here, there are different projects contributing to these results. So we should acknowledge these contributions. So thanks for joining today. Thanks, Gemma. Thanks a lot. And we'll meet in 2 weeks for a presentation on spectral representativeness applied to RTBs, which will be presented by Fabrice. So Fabrice, who is leading the work package 3 in RTB Foods, will be taking time to present this decision support method to identify the minimum number of spectra to be representative of the diversity of samples. So it will be a high throughput presentation, methodological presentation in 2 weeks. Fabrice, do you want to say a word to present and to invite people to join in 2 weeks, if you're still with us?
Yes, thank you. I'm still here, but it's a lot of pressure now. After a presentation like this, I hope I will be ready, but I should be ready. So it's just a way I will present some results, how to decide how many samples to do to be representative of the sample we want to analyze by NIR. So it's one of the way to do it, a easy way to try to make the decision to how many samples you have to do at minimum to be representative. So I will base my presentation on example on coffee, on cocoa, and on fruits, and what we have done in CIAT regarding cassava.
Okay, so it's not only applied to RTBs. For now, it has been applied to different crops.
It doesn't matter. It's a methodology, so it's not really. Sorry, yeah, it's not. Sorry, it's a methodology, so it could be applied to any kind of product. It's the same methodology.
Perfect. Okay, good. Thank you, Fabrice. So let's meet in 2 weeks. Yes.
Thank you, please.
Okay. So let's meet in 2 weeks, and until then, have a nice time. Take care, and have a nice weekend. Thanks, everybody. Goodbye.